Hello everybody, my name is Akshalova L M. I'm a teacher of biology of Nazarbayev Intellectual School in Astana. Today we will consider the very important topic in biological course. It is DNA replication. DNA replication is a very important process, especially during cell division, because new daughter cells should receive copy of genetic material exactly. DNA replication is a biological process that occurs in all living organisms and copies the exact DNA. It is a basis for biological inheritance. As you can see in this picture, parent cell copy genetic material, and after daughter cell receive each daughter cell receive copy of genetic material. DNA replication is a synthesis of daughter DNA from parental DNA. DNA replication occurs during mitotical or meiotical cycles. DNA replication occurs during the S phase of interphase. Exact copies of all the DNA on all the chromosomes are made. This gives sister chromatids, chromosomes, and their copy, which will be separated in the mitosis. When the cell divides through mitosis, each daughter cell therefore contains a full set of DNA. Mitosis ensures that each daughter cells gets the correct number of each chromosome through alignment at the equator. During metaphase, all chromosomes is located in the area of equator, and the separation of chromatids by spindle fibers during anaphase. We can see during anaphase, chromatids divide separated into two polar. When the cell divides during cytokinesis, there is a full set of chromosomes at each pole, forming new nuclei. The replication of DNA begins at a particular site called origin of replication, as short stretches of DNA having a specific sequence of nucleotides. Now here I'm drawing two DNA strands and label this. This site is origin of replication. Origin of replication reaches by adenine and thymine. Why adenine and thymine? We know about there is two hydrogen bonds between adenine and thymine, while between guanine and cytosine, three hydrogen bonds. There are three hydrogen bonds, and two hydrogen bonds are easily broken down. That's why origin of replication reached by adenine and thymine. The proteins that initiate the DNA replication recognize these sequences and attach to the DNA, separating two strands from each other and opening up the replication bubble. This place is called replication bubble. Replication bubble. At each end of replication bubble is called replication fork. Replication fork. This is a Y-shaped region where the parental strands are being unwound. Several enzymes participate in the DNA unwinding. First enzyme is helicase. I can label like this. This is helicase. Helicase are enzymes that untwist the double helix of DNA, separating two parental strands and making them available as template strands. Parental strand and second parental strand. After separating two parental strands of DNA, single strand binding proteins bind to the parental strand and keeping them from repairing. I can draw like this. This is single binding proteins. Single strand binding proteins. I'm repeating that these proteins keeping single strand of DNA from repairing. The untwisting of the double strand of DNA causes tighter twisting and strain of a replication fork. But there is another enzyme, topoisomerase, which helps relief by breaking, swiveling, and repairing DNA strands. The unbound sections of the parental strands are now available to serve as templates for synthesis of new complementary strands. However, enzymes which synthesize new DNA strands cannot initiate this process. They can only add nucleotides to an already existing chain. The initial chain, which is produced during synthesis, during DNA replication, is actually a short stretch of RNA, not DNA. This RNA chain is called primer. I can draw like this. This is short stretch is primer and synthesized by another enzyme which is called primase. Primase starts synthesize primer, short stretch of RNA from a single RNA nucleotide by adding RNA nucleotides at one time. The completed primer is for approximately 5 to 10 nucleotides long, and then DNA strands will start from adding DNA nucleotides to 3' end of primer. The adding of DNA nucleotides occurs by DNA polymerase. DNA polymerase is an enzyme which catalyzes the synthesis of new molecular DNA of DNA. DNA polymerase. There are several types of DNA polymerase, but only two types play the important role during DNA replication: DNA polymerase 1 and DNA polymerase 3. DNA polymerase 3 adds DNA nucleotides to the pre-existing chain. DNA polymerase 3 can add DNA nucleotides to the 3' end of primer. I can draw like this. This is polymerase 3. Add nucleotides to 3 end of primer and continues adding the DNA nucleotides to elongated end of new DNA strand. But we need to know that two ends of DNA strands are different. We know that one strand, for example, 3', second and 5'. And in addition, the two strands in a double helix of DNA are also different or anti-parallel. It means that two strands of DNA in double helix are oriented in opposite directions. For example, if one strand has direction from 3' to 5', second strand will have direction from 5' to 3'. It means that two strands of DNA are anti-parallel. And clearly that two new DNA strands also must be anti-parallel to each other. By the structure, DNA polymerase 3 can add DNA nucleotides only to the free 3' of primer and can add DNA nucleotides continuously. And along to one template strands, DNA polymerase 3 can make new DNA strand by adding DNA nucleotides continuously. And this new strand from 5' to 3' is made by this mechanism is called leading strand. Leading strand. To sum up, strand from 5' to 3', new strand along the parental strand from 3' to 5', always called leading strand because this strand will make continuously. To elongate the other of new DNA strand, we can see that DNA polymerase 3 must work away from replication fork in mandatory 5', 3' direction. What is the only one primer is required on the leading strand? Each Okazaki fragments must be primed separately. After polymerase 3 forms Okazaki fragments, another enzymes, polymerase 1, replaces RNA nucleotides of adjustment primer with DNA nucleotides. Replaces with DNA nucleotides, RNA nucleotides. But polymerase 1 cannot join Okazaki fragments together. This function will submit by another last enzyme, ligase. Ligase joins Okazaki segments together, and we can see that we receive second new strands of DNA. To sum up, we receive two new strands of DNA.
Cells like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of four nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
DNA replication mechanism. In results of this process, we can see that parental strand consists of the DNA consists of two original parent molecules. But after DNA replication, we can see new DNA molecule, but each DNA molecule consists of from one parental strand and second new strand. Also, second molecule has one old strand and second new strand. That's why this mechanism is received the name semi-conservative mechanism, because prefix semi, it means half. One strand save, second strand building. That's why semi-conservative mechanism. To sum up, main proteins assist in DNA replication. DNA helicases unwind the double helix. The template strands are stabilized by other proteins. Single-stranded DNA binding proteins make the template available. RNA primase catalyzes the synthesis of short RNA primers to which nucleotides are added. DNA polymerase III extends the strand in the 5' to 3' direction. DNA polymerase I degrades the RNA primer and replaces it with DNA, and last DNA ligase joins the DNA fragments into a continuous dotted strand. Enzyme in DNA replication: helicase unwinds parental double helix, binding protein stabilizes separate strands. Next, primase adds short primer to template strand. DNA polymerase III binds nucleotides to form new strands. DNA polymerase I removes RNA primer and inserts the correct bases. And last, ligase joins Okazaki fragments and seals other nucleotides in sugar phosphate backbone. Dear students, we have finished the theoretical part of our lessons, and now we will submit the practical work. Now we will work by online references by DNA replication. Please open these references. You can see this window. Use this science activity to learn about how DNA replicates. Unzip this DNA, then choose the correct enzymes to replicate the strand. After, students will work by this online activity. Here is a section of DNA that has unwound and is ready for replication. It needs for students click unzip and watch how an enzyme unzips the DNA. For example, when the DNA is ready to replicate, it unwinds into a straight double strand. An enzyme moves along the DNA double strand and breaks the bonds between its nucleotides, resulting in two separate single strands. The region of DNA where the strands separate is called replication fork. This place is called replication fork. DNA polymerase enzymes attached to the single strands help these enzymes make two new DNA strands by dragging and dropping the correct complementary nucleotide to each existing base where the DNA polymerase is attached. Notice that the new double strands are identical. For example, this process looks like it. For example, in the template of old straight, we can see guanine. Guanine complementary to cytosine. Next, for example, thymine. Thymine complementary to adenine, and the shape of adenine and thymine match to each other. Next, adenine complementary to thymine. Okay, cytosine complementary to guanine. Wait, sorry, guanine. Yes. Cytosine complementary to guanine. For example, if you will take thymine, no connection between these nucleotides. Guanine match to cytosine, thymine to adenine, and students will continue this process. And they can notice that new strands of DNA molecule grow. Guanine, thymine, adenine complementary, and guanine complementary to cytosine, and our strands are growing. Sorry, our process finished. We can see 2 new strands of new DNA molecule. Students can reset it. And next, our activity. Students can work by this worksheet for their formative assessment. First task, exploring DNA. First question, list at least 3 reasons why a cell must undergo division. Students can give different answers, but may include growth, repair, reproduction, the cells gets too big, surface area to volume ratio. Next question, imagine you are cutting a bagel, one of the most common household injuries, and you get a cut. The cut heals. How do the new cells compare to the original cells? Also, answer may vary, but may include exactly the same scar forms, cells are different age. And next question, how does your body ensure that the new cells are the same? Also, answer may include DNA contains the information in the old cells as well as the new cells. The DNA is the same in each cell. This question is very important for understanding the role DNA replication in the cell. And fourth question, how does DNA get into the new cells? Also here students can give different answers, but the answers can include, may not be accurate, but lead to discussion regarding DNA replication. These questions help to develop the skills of discussions. Next task, modeling DNA replication activity. In this task, they can learn how a copy of DNA is replicated for each cell, and they can create the model of DNA replication. Pair the nucleotides of the template strand of the template strands to the non-template strand of DNA. For example, here they should you construct it to create a double-strand DNA model. And tasks, record the template strand bases in the blank spaces provided above. These tasks should be very easy because they submit practical work before. They can submit. Next question, examine the strengths of DNA. What do you observe about the arrow ends of the model? Students can give answers. The arrows are on opposite ends of the strands. What does the arrow indicate? The arrow indicates the 3' hydroxy end of the DNA molecule. For example, we can see 3' end. Next task. Help to train work with DNA molecule and to create connections. For example, first task, examine the diagrams to the right, circle and label 3 carbon and 5 carbon in the DNA nucleotide. Here we can see 5' carbon in DNA nucleotides and 3 carbon. Primes are used in numbering carbons on this sugar portion of the nucleotide to distinguish them from the nitrogen-based carbons. In the image to the right, label the location of the 3 carbon and 5 carbon. Students should find 3' end and 5' end. How are the 3' and 5 carbons oriented in the strands of DNA molecule you assemble? Students notice that the 3 and 5' carbons are in opposite ends, or the two strands of DNA are anti-parallel to each other. This task help students to identify and label the replication bubble and replication fork in the model to the right. For example, this is replication bubble and displays replication forks. Looking at the mini tuber model, what do you think might be the first step of replication? And students should understand that the first step it is unwinding or separating DNA, and our molecule is becoming single strands. This task is connected with calculation. For example, nucleotides are added at an approximate rate of 50 nucleotides per second in eukaryotic cells. The human genome contains, for example, 6.4 billion nucleotides, which must be copied, and thus calculate the length of time in days that it would take to copy the human genome. And here we can see the process of calculation. Why do you think multiple replication bubbles form during the process of DNA replication? This task helps students for developing the critical thinking. For example, answer can be the replication process would be too slow if DNA replication occurred at a single bubble. Next question, what does the helicase appear to be doing? Helicase appears to be separating the two DNA strands, and identify which type of bond is broken. As students know about it, hydrogen bond between the nitrogen bases. And question, why is the helicase able to break these bonds? Students should demonstrate their knowledge that the helicase is an enzyme that facilitates breaking the hydrogen bonds between nucleotides. The pointed orange wedge in the helicase model represents the active site of the enzyme. This picture can help them to answer for this question. Yeah. Also, we need check the concepts, the understanding of concepts about leading and lagging strands. These questions, following questions, help for this process. For example, 21. As a new nucleotide is added to the growing DNA strand, which part of the new nucleotide forms a bond with the 3' OH group? For example, we can see 3' OH group from one old strand, template strand, and it will be the phosphate group. Sketch a helicase on the diagram to the right and indicate the directionality of the newly replicated leading strand of DNA. Students show leading strand because leading strand also always from 5' and to the 3' direction. Will you able to synthesize the other strand of DNA in a continuous manner when using the model? Explain why or why not. This question help students to develop the skills of explanation. For example, a student can give this explanation. DNA may be synthesized only in the 5' to 3' direction because DNA is the anti-parallel. The other strand would be synthesized in the 3' to 5' direction if it were a continuous synthesis. And leading strand, about lagging strand, sketch and indicate the directionality of the fragments composing the lagging strand of DNA right. Students should find the 5' end and 3' end of second template, and students should find replication fork and should demonstrate the direction of the process replication in opposite direction, because from 3' to 5' always replication process occurs in direction from 3' to 5'. And last questions. Why is DNA replication considered to be a semi-discontinuous process? This question helps to understand students the process of semi-conservation process, mechanism of DNA replication. Students can give this version. DNA may be synthesized only in the 5' to 3' direction. Because DNA is anti-parallel, the other strand will be synthesized in the 3', 5' direction if it were continuous synthesis. And in this picture, students need apply their knowledge and create a sketch which models in the semi-discontinuous processes of DNA replication at a replication bubble. Be sure to label the following aspects of your representation: leading and lagging strands, helicase, Okazaki fragments, parental strands, 3' ends and 5' ends. And students should draw the scheme like this. Maybe it will be another version, but similar it. How do these two new strands compare to the original parental strand? May include the fact that the two daughter molecules are identical to the parent molecule, that each daughter molecule is composed of half parental template DNA and half new DNA. And if students will have time, they can consider 3 models for the process of DNA replication and draw. For example, this model, students can compare 2 new strands and can notice that one strand consists of 2 new strands, and second molecule 2 strands of old molecule. This is conservative mechanism, and they can compare second experiment with previous experiment, and they can notice that each new molecule of DNA consists of from one old strand, black strands, and one new strands, red strands. That's why this mechanism is called semi-conservative, and this experiment show it. And finally, they using the colored DNA parental strands, they have just created in the gray nucleotides model the dispersive method of DNA replication, dispersive. And which of the methods can now be eliminated based on the results that Mendelssohn and Stahl got after one round of replication to Y? The version of answer, the conservative mechanism of DNA replication may be eliminated because it produces two bands in the density gradient test tube. Mendelssohn and Stahl experiment showed only one band after one round of replication. And they can compare three models of DNA replication: conservative, semi-conservative, and dispersive, and they should conclude that DNA replication occurs by semi-conservative model. And finally, the very important question: what is the replication of DNA? What is the relationship of DNA replication to cell division? DNA replication is the process by which cell make a copy of DNA for the daughter cell. It is very important answer for understanding this topic. And of the representation of DNA model identifies the strength and weakness of each. A student can give various answers. And finished. To sum up, we have studied the process DNA replication. I think we understood of the process of DNA replication, the mechanism of DNA replication, and the role of this process in the cell's level. Thank you very much for your attention.